Hey guys, in this video, we're going to continue talking about populations. In this very short video, I am specifically going to talk about a couple methods that ecologists use to estimate the size of a population, specifically populations that essentially stay in one place. Plant populations in particular. So one way that ecologists can estimate the size of a population and therefore calculate cool things like how quickly the population is growing or declining, and therefore predicting essentially how the population is going to change in the future. We use what are called quadrats. So essentially taking a single size, in this case a 1 meter by 1 meter square. It can be larger than this, but 1 meter by 1 meter square, and essentially counting every single organism of your species of interest within that space. You might do this a couple times or just once, and then use that number to make the other calculations. In class together and or at home, we are going to be practicing making quadrats and then counting all of one particular species inside that quadrat, and then kind of playing with those numbers a little bit. Okay, so I will show you this video of a previous class at Stockton working with a quadrat.
So this is a quadrat. These guys are counting, it looks like, wild blueberry within a meter by meter square. So this is also what you guys are going to do. Get, approximately 1 meter square spot and count one particular type of plant within it.
And so we will do that. Another method that is used to estimate the size of plant population, so how many plants there are in the entire population, is something called a transect. And so this is where you draw a straight line through an area and you count all of the organisms of interest on either side within a particular buffer region, or you can even go further than that and measure different characteristics about them. So what we're going to see here is, again, a previous ecology class measuring the height of oak trees. So measuring the height, you can also measure kind of the circumference of the tree, so getting kind of a good idea of how big the trees are in the area, maybe like an age class kind of a thing. You can measure how many trees are infected by a beetle. Like whatever your question is, you can measure along this transect and get a pretty good idea of what is happening in the entire forest. And so let's watch this class.
Here we have section 2B. There's a transect to make a straight line through the forest. And here we have the measuring of a single oak tree.
Okay, so in our class, we may do this as well. Again, pretty good sampling of a population. Another way that you can estimate the size of a plant population, or the number of a population that is experiencing a particular condition or has a particular phenotype, is through different types of imaging. So here we can see aerial imaging, so we can, if we are trained in recognizing these different species, we can actually go through this image and pick out all of the pine trees versus other types of trees. Of course, this is very difficult, but this is where special types of imaging technology, such as using that infrared technology, can really help out. I also wanted to make a side note here that these types of methods are great for measuring anything that doesn't move. So this can also be animals, right? So here we can see barnacles, they don't move, and so we can really easily just calculate the area of this mass and then count all the little barnacles within it, and then we can use that to estimate the size of the entire population. And we can do this with corals, we can do this with anything that is immobile. Okay, so once we have our little sample, right? So 1 meter by 1 meter or 1 kilometer by 1 kilometer, or whatever, we can find the density, right? So the number of organisms in a particular space. Okay, we talked before about crude density versus ecological density. The only difference really being ecological density does not consider spaces where that particular organism is not located. Okay, so if we want the entire population, we of course find the density of our samples and then use that density, right? 10 blueberry bushes per meter squared times the entire range area. So maybe we're thinking about, what is the entire population of blueberries, wild blueberries within the entire pine barrens. And so then we could use that density calculated from our little quadrat, multiply it by the area of the entire pine barrens to get our estimate of the plant population. Thank you guys so much. Stay tuned for our next part of the lesson.